Hey everyone, welcome back to Neurobiology at Providence College. I'm Joe DeGeorgis. We're going to do another pretty short paper today, one that I think is really cool and has a really important element to it, which I'll get to in the end. The paper's by Myerowtz and Costigan. The title of the paper is "The Major Defect in Ashkenazi Jews in Tay-Sachs Disease is an Insertion in the Gene for the Alpha Chain of Beta-hexaamidase." I don't think much is known about Tay-Sachs disease other than the fact that a mutation in this particular gene for beta-hexaamidase causes the disease. It's an autosomal recessive disorder, and the enzyme itself is in the lysosomal pathway, and it looks like it's involved in some sort of fat metabolism. And they say that there's a fatty type of tissue that builds up in the brain and that kills the nerve cells. And there's a very early onset to this disease. It often occurs as early as 6 months of age, and most children afflicted die by the age of 4. So it's a childhood disorder. It turns out it occurs frequently in Ashkenazi Jews because of inbreeding, essentially. I mean, members of that community want to marry and have families with other people in that same community, which is, of course, understandable and extremely common in all ethnic communities. And because of that, the disease is propagated within that group at a frequency that's about 10 times higher than the overall population. Okay, so let's take a look at what they did in this paper. First of all, they say that this protein consists of both an alpha subunit, It has both an alpha subunit and a beta subunit. So that means two different proteins, that is, two different gene products, and those produce beta hexaamidase. Okay, so don't get confused by this beta and this beta. There's a beta subunit of the beta hexaamidase, and there's an alpha subunit of the beta hexaamidase. And it turns out that in studying the population of Ashkenazi Jews, they thought that the mutation was a single mutation in the alpha subunit, and that was due to a founder that had a mutation, that is, an individual many generations ago, 10 generations ago, 20 generations ago, had a sporadic mutation, and that mutation caused all of the individuals in the Ashkenazi Jewish population to carry that gene that causes the disease. And it turns out that they ultimately found a mutation in the alpha subunit that altered the splice junction of the gene. So we've talked about splicing before, and of course you have double-stranded DNA in the genome, which goes on for a long ways in both directions on the chromosome, and one of the strands is copied into an RNA molecule that's identical to the top strand here, except the U's, of course, are added instead of T's because it's an RNA molecule. And then in this particular RNA, which is not a mature RNA, but something called a heteronuclear RNA, which we've talked about before, heteronuclear RNA, contains both introns and exons. So one region, like this region here, might be an intron, which is an intervening sequence, intron 1, and then you have a region that is an exon, and then another intron, and an exon, 2, and so on. And when the RNA is spliced, these intervening sequences get removed, and the exons are spliced together so that you have exon 1 2, 3, and so on. And then ultimately a poly A tail is added to the RNA, and there's a 5' cap. And of course, somewhere within this region, there is the start site of the coding sequence and the stop codon, which terminates translation. And this part here, of course, is the coding sequence that gives rise to the amino acid sequence, or encodes the final amino acid sequence. Okay, you guys, I have to correct my mistake here. Obviously, the start of the coding sequence isn't here, but is here. It starts at the first ATG that encodes the methionine, and then it ends at the codon, or the triplet nucleotide sequence that encodes the last amino acid, which is just prior to the 3 nucleotides, or the codon, if you will, that encodes this particular stop codon. So what they're trying to say is that there's some problem that they've detected in the splicing of this particular sequence, or a sequence in between an intron and an exon. And so what that means is that the splicing didn't happen, and that the intron ultimately was inserted here and was still in between exon 1 and exon 2. So when you get to the end of exon 1 and you're reading 3 at a time and translating that into the amino acid, when it gets to the end of exon 1, it starts to read the intron, which of course is incorrect, and most of the time that results in an early termination signal and gives you a truncated form of the protein. So at any rate, they thought that this particular mutation was responsible for all heritable forms of Tay-Sachs in the Ashkenazi Jewish population, and so they tested individuals that had Tay-Sachs within that community, and it turns out that's not true, that only about 20 to 30% of individuals in that community that have Tay-Sachs disease can be explained by this particular mutation. So it means that 70 to 80% of the Tay-Sachs individuals in that community must have some other mutation. Okay, so what did they do? Well, they took an individual in the Ashkenazi Jewish community who was afflicted with Tay-Sachs disease, but did not have this particular mutation, and they sequenced that individual's gene for the alpha subunit of beta hexaminidase. When they did that, they found a new mutation that was a 4-nucleotide insert into one of the exons, which was exon 11. Then when they tested members of the community, they found that individuals that were carriers had this particular mutation 70% of the time, which we're going to get to the test in a moment. So it looks like this mutation is the major mutation that causes Tay-Sachs disease within this particular community. Okay, let's talk about their assay. In this paper, they used a DNA dot blot. So in some ways, it's similar to that used in the fatal familial insomnia paper. However, in that paper, they were looking at protein. And you'll recall that they were looking for protease-resistant forms of prion. They purified the protein, they added proteinase K, and then they measured whether the protein was still present, that is resistant to protease K, or not resistant, by using an antibody that binds to the protein in the dot blot, and then a secondary antibody that changes color. So for DNA, you can't use an antibody. You need to use a radioactive or some sort of colorimetric form of DNA. So they built these probes, and I'm going to try to take you through this process. If you have a wild type individual they of course lack this 4 base pair insert, which is the mutated form. So I'll write that over here and save that for a moment. And the wild type form of the gene, this is exon 11 of the alpha subunit. These two in a wild type individual are of course normal, and they lack that 4 base pair insertion. So if we look now at an individual that is afflicted, that is, they have 2 copies, since this is a recessive disorder, they will have 2 copies of the 4 base pair insert, and this wild type individual will lack that inserted form. But you can make PCR primers that flank the 4 base pair insert, so would bind here and here, and of course the same here and here. And those same primers will also bind to the same sites in the wild type form, but the 4 base pair insert doesn't exist in this particular case. So in wild type, when you PCR amplify the genomic DNA of the wild type individual, you're going to get two PCR products that are from here to here in length, and they're both going to be identical. So the one amplified from this chromosome, the first allele, will be of this size, and the one amplified by the second allele will be of this size. And of course, it's a PCR product, so this is double-stranded DNA. When you amplify the DNA from an individual who's afflicted with the molecule that has the 4 base pair inserts, well, you're going to do the exact same thing, and you're going to amplify the DNA sequence that's in between and including the PCR primers. So here, and then another one. And these are identical in this case, and these two are identical in this case, but this one has the 4 base pair insert. And what they do is the PCR reaction in the presence of radioactive nucleotides. So that they're hot. They don't all have to be radioactive, just one could be radioactive, but that's going to make these probes radioactive, and in the end, in a few moments I'll explain, they lay a piece of film, photographic film, over the dot blot, and the radioactivity exposes the film, and then when you develop the film, you see the radioactivity, whether there was radioactivity there or not. But let me get to that in a moment. Okay, let's talk about the dot blot. Again, we're using a membrane. This in the case of DNA, it's a little bit different than the nitrocellulose, but it's a similar type of material. It's just kind of a paper type of stuff, and you can cut it like a piece of paper into strips. And they have 2 probes that they're going to use to test individuals as to whether they have the 4 base pair insert mutation or whether their allele is wild type, or one of each. So they call one the normal probe, which of course is without the 4 base pair. And then they have one called mutant probe. And now they take the DNA from different individuals, and they purify the DNA, and they heat it up, and that separates the 2 strands so that we have individual molecules. And in the case of, so if this is a strip of of wild type 1, the top strand of wild type, and the bottom strand of the wild type chromosome that goes on for a very long time, then the wild type primer that we developed, which is now radioactive, can also be heated and denatured so that they're individual strands, and now this strand can bind to this piece of DNA, and this strand can bind to this piece of DNA. And the same is true for the mutant probe, except the mutant probe won't bind to wild type DNA. It will only bind to mutant forms of the DNA strands. Okay, you guys, my computer decided to crash. Oops. So look, let me just redraw the blot here. So what I was trying to get at is that the probe developed against the wild type form will only bind to the wild type DNA if the individual has that form of DNA, and the mutant probe will only bind to the mutant form, right? I mean, it makes sense. So this is normal, again, sorry about that, probe. This is mutant probe, which has the 4 base pair insert. And ultimately, like I mentioned a few moments ago, after you dot blot the DNA onto the piece of nitrocellulose, you have to incubate it in a solution with the probe, and the probe finds its complement, if it exists, in the dot, and then you put a piece, you wash off the solution, the excess solution, you put a piece of film over the top, and the radioactivity exposes the piece of film and then colors the film dark if indeed the probe bound to the DNA. So they have different individuals. They have individual 90, which is a wild type individual that's not of Jewish descent. So I'm just going to write wild type here. And then they have an individual who is Jewish, and they are called SW, and they are also wild type, or normal, however you want to say it. So I'm going to put wild type up here. Then we have an individual who is Jewish, and they are a carrier of this 4 base pair mutation. So in theory, this person is heterozygous and should have 1 normal copy and 1 mutant copy, which we'll get to in a second. Then there's an individual named RB, and they are afflicted. So I'm just going to draw a circle with a line through it for afflicted. Then there's TB, who is a carrier and is the father. I'll write C for carrier, but this person is the father of RB. Okay, the father of RB. Then we have BB, who is the mother of RB, and she is also an unafflicted carrier. Then we have 2968, who is afflicted. So I'm just going to abbreviate this 68. And then we have 52, who's this individual 68's mom. We have 51, who's the father. And then we have an individual who is 505, which is another individual who is afflicted. This is a carrier. This should be a carrier. Now, if we take a look at the dot plot in the paper, number 90 is a wild-type individual who is not of Jewish descent. So if you look at the dot plot, you see that they have wild-type DNA for the wild-type or the normal probe, and they lack any dot plot in the mutated probe. If you look at individual SW who is of Jewish descent, and is wild type, then they also have the same pattern. Now SS is an Ashkenazi Jew, Jewish person, and they are a carrier, they say, for this probe, and so they have a wild type form and they have a mutated form, and then we have an afflicted individual, and the afflicted individual only this is RB only has the mutated form, and they're afflicted, so it means they must have two copies. It's an autosomal recessive disorder, so that individual has it. RB's dad is a carrier, so he has one of each. Cool. Mom is a carrier, so that explains that issue. Number 68 is an individual who's afflicted, but interestingly enough, they have one copy that the wild type probe picks up and one probe that's picked up by the mutation. So they only have one copy of the 4 base pair insertion, and they have one that's detected by the normal probe. 68's mother is a carrier, so she, it turns out, has one copy of a wild type or something detected by the wild type probe, and one copy detected by the mutated probe. And then number 68's father has something detected by the normal probe, but nothing detected by the mutated probe. An individual 505 has one of each, and they are afflicted, so they only have one copy of the 400 base pair band. Okay, this makes sense. It's a wild type individual and they are just detected by the normal probe. Same with this particular individual. This person is a carrier, so they only have one copy of that 4 base pair insertion. This person has 2 copies, so they're afflicted. All of that makes sense. The father is a carrier, so this RB received one bad copy from the father and one bad copy from the mother, and all of that makes sense. The problem arises when you start to look at these 4 individuals. But remember, there are 2 different mutations that have been discovered in the Ashkenazi Jewish population. This one here happens to be the 4 base pair insert. Here, 4 base pair insert. But the first mutation that was identified was the splice site mutation that we talked about earlier in the lecture. And so they tested these individuals for the presence of the splice site mutation. Now, it turns out that individual 68 obviously gets 1 copy of the 4 base pair insert from that individual's mother, and so the other allele must be coming from the father, who shows normal probe in this particular case, but he tests positive for the splice site mutation, which isn't detected in this assay. It's a different assay. So both of his copies of the gene are normal at that particular region of the molecule. That is to say, the normal probe binds to 2 normal parts of the gene, and the splice site mutation needed to be assayed through a different approach. They then assayed individual 68, who's an offspring of 51, and they test positive for the splice site variant. This is splice, and this is 4 base pair, but it's both in the same gene, and ultimately that gives them a double mutant at that locus, at that allele, at that gene, and that individual is afflicted. Okay, so the 2 controls, wild-type controls, are explained by the fact that they only react to the normal probe. These 3 individuals are explained by the 4 base pair probe, either as carriers or afflicted, as in the case of RB. And these 3 individuals, 68, 52, and 51, are a mixture of the 2 different mutations that have been discovered within the Jewish community, the 4 base pair insert and then the splice site mutation. The real question came with individual 505, who has 1 copy of the 4 base pair mutation, and when this individual was tested for the splice site variation, because they have 1 copy that interacts with the normal probe, this person lacked the splice site mutation. So, since this individual is afflicted, they must have 2 bad copies of that particular gene. Here's 1 copy, and this one also must be mutated, which suggests that there must be a 3rd mutation in that particular gene that causes the disease in this particular individual. For sure, this group eventually figured out what that 3rd mutation is, because it's not that hard of an experiment, and it's an interesting finding that would give them another publication. They then went on to test to see whether they could detect protein from the 4 base pair mutant, and it turns out that they were unable to detect a protein product from that RNA molecule, and they believe that that is somehow due to a low expression of the RNA itself. So that's also an interesting phenomenon. But here's what I think is cool and why I like the paper so much. Because they've now been able to develop 2 different tests, one that covers 70% of the cases in the Jewish population, and another mutation that covers 20 to 30%, they can very easily, with this dot blot test or the splice mutant test, figure out who the individuals are who are carriers in the Ashkenazi Jewish community, and they've just used genetic counseling to try to advise people that are carriers not to end up having children together to increase the likelihood of the double mutation. And it turns out that the population has been so successful at doing that that they've lowered the incidence of Tay-Sachs in their community to rates that are lower than they are in the general population. And if two people who are carriers want to have children, that's fine too, except that they're well informed and they know, kind of what they're up against. So it's just a really nice story, I think. And I wonder if it reminds you of anything, since there's no cure and there's no real medication or anything like that that can even alleviate much of the symptoms. You can just alter your behavior to try to solve the problem. And it reminds me of what we're going through now, which is social distancing. Because, hey, guess what? We don't have a cure and we don't have a magic bullet to stop the virus from spreading. So, social distancing is the key for now. Okay, you guys, well, that was a little longer than I had expected because it's kind of a short paper, but the technique is, I think, pretty interesting, a little bit more complicated, or took more of an explanation than I thought. But anyway, that's it for today. I'll talk to you guys tomorrow. Thank you.